Welcome everyone. Today we are going to learn about ATP driven pump. Now at first I want to mention that there are three major categories of transport protein is present. One is channels, second is transporter. Under the transporter there are uniporters, symporters and antiporters. And the third category is ATP driven pump. Now ATP driven pump contains four different class of proteins. One is called P class pump, V class pump, F class pump and ABC transport. In this video, we are going to learn about ABC transporters. The full form of ABC is ATP binding cassette. It is also called ABC superfamily. The question is why it is called superfamily? Because it is the largest and most diverse family of ATP driven pumps. Now in my previous videos on V class pump, P class pump and F class pump, I have mentioned that all these pumps are actually transport only ions. Now in case of ABC superfamily, they not only transport ions, some member of the ABC transporter or pump actually transport ions, but they can also transport amino acids, peptides, even proteins that can be larger than the transporter itself. Sugars, lipids, cholesterol, bile acids, drugs, toxins, etc. But one thing we have to understand that don't confuse that one ABC transporter can transport all these molecules. No, there are different ABC transporters and one ABC transporter can actually transport one kind of or one type of molecules. Suppose if one ABC transporter can transport amino acids, it could transport peptides or even proteins, but it will not transport sugars, lipids or drugs. Okay, so remember this. Now this ABC transporter or ABC superfamily molecules are present in the bacterial plasma membrane, in the mammalian plasma membrane and the endoplasmic reticulum membrane. One thing I want to mention in case of E. coli bacteria, as because there is two layer of membrane is present, in case of E. coli bacteria, the ABC transporter is present in the inner bacterial membrane. Now all the ABC transporter, whether it is present in the prokaryotes or eukaryotes, they consist of two transmembrane domain we call T domain and two ATP binding domain or A domain. As you can see, this is an example of an eukaryotic ABC transporters. Here it is showing that this ABC transporter is actually exporting the molecule, and in this case there is two conformations. One is called inward facing, where the molecule binds with this ABC transporter, and outward facing one conformations that in which the molecules actually export occur. Now ATP actually when binds, the conformation actually changes from inward to outward, and when ATP degradation or hydrolysis occur, it actually converts into another time into the inward facing configurations. The first eukaryotic ABC protein that was discovered during the studies on tumor cells, because these tumor cells that express this ABC transporter show multi-drug resistance to the chemotherapeutic drugs. So that is why it is called MDR1. In latter it is called ABCB1. As you can see, when toxic drugs or chemotherapy drugs are treated with these cells, most of the cells actually die, but some cells that actually express high amount of this ABC transporter, that's why it is called multi-drug resistance, because they can actually pumps the toxic drug out of the cells, and then these resistant cells actually grow and expand the clone, so it actually hamper the chemotherapeutic treatment of the cancer. Now in contrast to bacterial ABC proteins, which are built of four discrete subunits, because there is two T domains and two ATP binding domains, all four domains of ABCB1 are fused into a single protein. As you can see, this is example of prokaryotic ABC transporter. Here two T domains and two A domains are actually separate subunits, but if you see the MDR, that means the MDR1 or ABCB1, you will find that all the subunits actually linked with each other, and it makes or generate a single protein. The size of the protein is 170 kilodalton basically. Now this ABC transporters expressed in abundance in the liver, intestines and kidneys. These are the sites of where toxins are actually secreted out of the body. Now an ABC transporter called transporter associated with antigen processing, or in short we call TAP, present in most vertebrate cells in the rough endoplasmic reticulum membrane. As you can see, there is two TAP proteins called TAP1 and TAP2 present. These are the ATP binding domain and these are the T domains, that it means transmembrane domain.
What happens in normal cells of every vertebrate normal cells, protein turnover occur. Protein turnover means after the half-life of the protein, the protein is degraded by proteasomes, and it actually becomes peptide fragments, and through the tap protein it enters the rough endoplasmic reticulum, and these peptides are actually loaded onto the MSC class 1 molecules, and then it is represented to the cells on the plasma membrane and scrutinized by CD8 cell or cytotoxic T cells. But in case of if the vertebrate cells is infected by virus, or if the cells is transformed into a tumor cells, then the protein is actually, in case of viral infections, the proteins are actually degraded by not only a proteasome but a modified proteasome called immunoproteasome. Then these peptides similarly enters through the tap proteins into the rough endoplasmic lumen, where it is loaded on the MSC class 1, and this MSC1 actually goes to the plasma membrane and then scrutinized by the CD8 cell. We call this CD8 cell cytotoxic T lymphocytes. If the cells is normal, healthy, then it will not kill the cell, but if it is virally infected, then this cytotoxic T lymphocyte can recognize that this is not a body antigen or self antigen, it will kill the cells. Also, in case of tumor cells, as because there is any mutation occurs in the protein, so it is called neoantigens. Cytotoxic T lymphocytes can also recognize that this protein is not normal, it is neoantigen, then also the cytotoxic T lymphocyte can kill. Now, another important point is that ABC proteins can act as flippage. Now, what is flippage? In my plasma membrane video, I have mentioned that phospholipids can perform 4 different movements. One of them is called flip-flop, and that movement occurs very rarely because this movement is actually energetically unfavorable. Now, there is some enzymes called flippage, floppage, or scramblage. One of the enzyme called flippage can move the phospholipids from cytosolic leaflet to the outer leaflet. So in case of eukaryotes, there is a ABC proteins present called ABCB4. Another way we can say MDR2, that is present in the liver cells plasma membrane that face the bile canaliculi. They actually release the phosphatidylcholine, one type of phosphoglycerides, from cytosolic leaflet to the exoplasmic leaflet, because flip-flop movement means movement of these phospholipids from one leaflet to the other leaflet. Now, as we know that plasma membrane made up of 2 lipid bilayers, so the movement or migration of one phospholipid from one layer to the other layer is called flip-flop movement. So in case of flippage, this ABCB4 actually moves the phosphatidylcholine from the cytosolic leaflet to the exoplasmic leaflet and ultimately into the bile. Now there are also other different ABC proteins present that also transport cholesterol and bile acids to make the bile. Hope this video will help you. If you have any doubt about this topic, you can write me in the comment section. Thank you for watching, and always keep learning.